Today I'll be talking about the role of S100A8 positive macrophages in the development of steatotic liver disease, especially in the context of adipocyte death. So the first slide shows the progression of metabolic dysfunction associated steatotic liver disease, or MESLD, which comprises a couple of metabolic liver diseases ranging from steatosis to MASH fibrosis and hepatocellular carcinoma. Steatosis is characterized by an excessive fat accumulation in the liver, and steatosis may further progress to MASH, which now has inflammation and fibrosis in addition to fat accumulation. MASH can further progress to cirrhosis and carcinoma in an irreversible manner. So researchers are trying to understand the mechanism by which steatosis progresses to MASH and target that mechanisms for the therapeutic intervention. I think many of you in this room know that metabolic syndrome is an important risk factor for the development of MESLD. And obesity is apparently the most remarkable features of metabolic syndrome. So if you imagine a person with obesity, you may think about the big belly. Inside the big belly, what's going on? So in obese individuals, adipocyte death is frequently observed, which is associated with unhealthy expansion of adipose tissue. So when adipocytes undergo cell death, they release free fatty acids, which may come to the liver, especially in hepatocytes, which is this green rectangle. So in the hepatocytes, free fatty acids may cause lipotoxicity, which causes multiple types of stresses in cells, like ER stress, oxidative stress, and other types of stresses. And they eventually lead to the death of hepatocytes. And during that kind of process, cytokines and chemokines are released, which may further enhance inflammation and fibrosis. So lipotoxicity is an important kind of contributing factor for the development of MESLD. So there are many types of cell death, of course, in adipocytes as well. And among those many types of cell death, researchers have reported the involvement of apoptosis in obesity. So this paper published in 2010 reported that adipocyte apoptosis contributes to adipose tissue macrophage infiltration and insulin resistance associated with obesity. And in another study conducted in human samples, it has been reported that caspases like caspase 3 and 9 are upregulated in adipose tissues of obese individuals, while BCL2, which is an anti-apoptotic factor, is downregulated in adipose tissues of obese individuals. So here's a brief kind of explanation for apoptosis pathway. So BCL2 and BCL-XL are representative anti-apoptotic factors which block the release of cytochrome C from the mitochondria, thereby inhibiting the execution of apoptosis. In contrast, caspases 3 and 9 are crucial for the execution of apoptosis. So this slide shows an important kind of association between adipocyte apoptosis and obesity. When our laboratory fed mice with a high-fat diet for up to 4 months to induce steatosis in the liver, we observed in adipose tissues an interesting kind of phenotype. So this signal is paralipin expression. Paralipin is a protein which is expressed on the surface of adipocytes. So if adipocyte undergoes cell death, adipocytes lose the expression of paralipin. So after 3 months of high-fat diet feeding, we found a reduction in the expression of paralipin, indicating adipocyte death. And the death of adipocytes was further confirmed by tunnel staining, which also became kind of pronounced after 3 months of high-fat feeding. We also looked at the markers for adipose tissue inflammation, such as crown-like structure formation and macrophage infiltration. Both of them also became pronounced after 3 months of high-fat feeding. And we also, of course, looked at the liver. In the liver, fat accumulation and macrophage infiltration started to increase at 3 months, which is identical to the period when adipocyte death started to occur, which we just saw in the previous slide. And inflammatory and fibrotic gene expression also started to increase at 3 months. So if we put these things together, what you can see is, so 3 months and 4 months of high-fat feeding reduced the weight of epididymal fat tissue in mice. But before that period, until 2 months of high-fat feeding, the weight of adipose tissue became elevated. So we can say that there was a shrinkage in epididymal fat mass after 3 months and 4 months of high-fat feeding. And at that moment, adipocyte death became pronounced. And okay, in the liver, until 2 months, there was not much increase of liver weight. Also, there was not much increase of liver fat contents. But after 3 months of high-fat feeding, it started to be elevated. So there is a temporal correlation between adipocyte death and hepatic fat accumulation in obese mice. So we became interested in the involvement of adipocyte apoptosis in the temporal correlation between the two organs, which are adipose tissue and the liver. So as I mentioned earlier, BCL2 is an anti-apoptotic factor, and it is downregulated in adipose tissues of obese individuals in humans. So we hypothesized that if we could overexpress BCL2 specifically in adipocytes, we can inhibit adipocyte death and attenuate adipose tissue inflammation. So to prove this hypothesis, we established adipocyte-specific BCL2 transgenic mice by crossing adiponectin-CRE mice with knockin mice, which express the BCL2 gene coding sequence, which is also preceded by stop codons that are flanked by LOXP sites. So by crossing with adiponectin-CRE, we can delete the stop codons, which initially inhibit the expression of BCL2. So we expected them to overexpress BCL2 in adipocytes. So we characterized these animals, and we found that the expression of BCL2 was indeed increased in the liver, in the adipose tissues of the transgenic mice at both protein levels and messenger RNA levels. Also, I said earlier that after 3 months and 4 months of high-fat feeding, actually the size of epididymal fat tissue became reduced, which I mentioned as shrinkage of adipose tissues. But in transgenic mice, actually that kind of shrinkage was protected. So apparently the weight of adipose tissues was greater than that of wild-type animals. So also we could kind of obtain this positive correlation between BCL2 messenger RNA levels in adipose tissues and the size of epididymal fat tissue. So if you overexpress BCL2, you may inhibit the death of adipocytes, thereby inhibiting the shrinkage of adipose tissues. And that can be summarized like this in this graph. We also measured the loss of paralipin by immunohistochemical analysis, indicating that in transgenic mice, the loss of paralipin was protected. So this indicates that adipocyte-specific BCL2 overexpression attenuates adipocyte death in obese animals. And then we looked at the markers for inflammation, such as crown-like structure formation and macrophage infiltration. And you can see that in transgenic mice, the elevation of macrophage infiltration was inhibited, which could be also further confirmed by flow cytometric analysis and real-time qPCR. So to summarize the first part, we found that BCL2 overexpression specifically in adipocytes inhibited adipocyte death and adipose inflammation. Then we became interested whether liver could be also affected by the modulation of manipulation of BCL2 gene in adipocytes. So we looked at the lipid deposition in these transgenic animals in comparison to wild-type animals. And you can see that lipid deposition was reduced in transgenic mice, which could be also confirmed by biochemical analysis of lipid triglyceride contents in the liver. Also lipogenic genes such as fatty acid synthase and fatty acid uptake-related gene CD36 were downregulated in the transgenic mice. So adipocyte-specific BCL2 overexpression attenuated high-fat-induced hepatic fat accumulation. We also looked at the markers for kind of liver injury, such as ALT levels. So in transgenic mice, ALT levels were also reduced, indicating that high-fat-induced liver injury was protected by overexpression of BCL2, specifically in adipocytes. And we also could obtain this kind of negative correlation between ALT levels and BCL2 expression, also ALT levels and epididymal fat mass or weight. So this indicates that adipocyte-specific overexpression of an anti-apoptotic factor, BCL2, reduced high-fat diet-induced liver injury. So this kind of result was very fascinating, and we moved on to study the cellular mechanisms by which adipocyte apoptosis modulates steatosis. So to address this question, we performed single-cell RNA sequencing on liver cells from BCL2 transgenic mice and wild-type mice, and we could find the unique subset of macrophages that express S100A8, which is kind of designated as cluster 6 here. So, okay, so unique subset of S8 positive macrophages was significantly enriched in wild-type mice in comparison to the transgenic mice. And that was further confirmed by histological analysis in wild-type mice and BCL2 transgenic mice. And you can see that IBA1 positive, S100A8 positive macrophages, the population of them, their population was apparently reduced in the transgenic mice. So adipocyte apoptosis may induce the infiltration of S8 positive macrophages in the liver. And that was further confirmed in human samples. So in patients with MASH, the population of S8 positive macrophages was elevated, as you can see here. And then we asked whether indeed adipocyte apoptosis can increase the expression of S100A8 in macrophages. So we performed an in vitro experiment shown here. So in this experiment, adipocytes were treated with or without phospholigand that induces apoptosis, and we collected condition media from these conditions and treated bone marrow-derived macrophages with the condition media. And we found that the expression of S100A8 in macrophages was elevated after the treatment with the condition media obtained from phospholigand treated, which means apoptotic adipocytes. Then we asked what in this condition media mediates S8 induction in macrophages? So the first guess was free fatty acids. As I mentioned in the previous slides, adipocyte death releases free fatty acids. So we confirmed whether free fatty acid is elevated after the treatment with phospholigand. And indeed, the levels of free fatty acids were elevated in the media of the cultures in the presence of fast ligand. And then we directly treated the macrophages with palmitic acid, which is one of the representative free fatty acids. And you can see that palmitic acid treatment indeed increased S8 expression in macrophages, which is similar to here. Then the next question was how S8 positive macrophages contribute to fat accumulation in hepatocytes. So as I mentioned earlier, CD36 was reduced in BCL2 transgenic mice. And we became interested in the role of CD36 in mediating the S8 positive macrophages induced fat accumulation in hepatocytes. So high-fat diet feeding for 3 months, as I said earlier, induces adipocyte death, while 2 months of high-fat feeding does not. And you can see that CD36 expression in hepatocytes was elevated after 3 months of high-fat feeding, in contrast to 2 months of high-fat feeding. And also, if we block the apoptosis in adipocytes by overexpression of BCL2, we could also attenuate the expression of CD36 in hepatocytes. So this means that adipocyte death may trigger CD36 induction in the liver. And then we moved on to examine whether S8 positive macrophages induce CD36 in expression. So to answer this question, we performed another co-culture experiment in which bone marrow-derived macrophages were treated with or without palmitic acid, and condition media was collected, which were added into the culture of AML12 cells. And as I mentioned earlier, palmitic acid treatment actually increases the expression of S8 in macrophages. So palmitic acid treated macrophages, which can be called as S8 positive macrophages, release something that could be transferred to hepatocytes, and that led to the elevation of CD36. And this effect was reversed by S8 knockdown in macrophages. So this indicates that S8 positive macrophages contribute to adipocyte apoptosis-associated CD36 expression in hepatocytes. And then we wanted to look at the in vivo effect of S8 positive macrophages in steatosis. So we established macrophage-specific S8 knockout mice and subjected them to high-fat feeding. And we could kind of get a promising result as shown here. So the liver weight and ALT levels were reduced in knockout mice. Also lipid deposition was reduced. And CD36 expression was reduced in the knockout mice. So macrophage-specific deletion of S8 gene led to less steatosis in mice. And then we wanted to figure out the molecular mechanism behind this process. So we performed bulk RNA sequencing with knockout mice and wild-type mice, and we could find 182 upregulated genes. If you zoom in, you can see these two genes, CCN3 and 5. These have very similar names, and that's why we became interested in these two genes, whether these two genes are really involved in this process. So we first tested whether palmitic acid treatment can indeed downregulate CCN3 or CCN5. And you can see that CCN3 and 5 expressions were reduced upon palmitic acid treatment. However, this kind of regulation was reversed when the cells were knocked down with S8. So this means that palmitic acid treatment suppressed the expression of CCN3 and 5 in an S8-dependent manner. And then we studied the role of CCN3 and 5 in CD36 regulation. To do this, we first performed literature analysis to see whether there is a kind of previous reports on the role of CCN3 or 5 in the regulation of CD36. And we could find this kind of information. So CCN3 knockout increased CD36 expression in aortas, but there was no report on the role of these in the liver. So we directly treated hepatocytes with CCN3 and 5, and we found that only CCN3 could reduce the expression of CD36 in AML12 hepatocytes. And we also looked at the upstream regulator of CD36, which is PPR gamma. CCN3 treatment reduced the expression of PPR gamma as well. And then lastly, we performed this experiment to see whether conditioned media obtained from palmitic acid treated macrophages, which are SA positive macrophages, could modulate the expression of CD36. And indeed, that was increased. And additional treatment with CCN3 actually reduced the induction of CD36 caused by palmitic acid treated macrophage conditioned media. So this means that SA positive macrophages induce CD36 in hepatocytes by suppressing the release of CCN3. Okay, so here is the conclusion of the today's presentation. So we found that adipocyte death is temporally correlated with hepatic fat deposition and injury in high-fat mice, which is kind of initiating information that we dive into this kind of research. By utilizing BCL2 transgenic mice, we found that adipocyte apoptosis promotes steatosis and is associated with the increase of SA positive macrophages in the liver of high-fat mice. And we also found that hepatic steatosis was attenuated in macrophage-specific S8 knockout mice. Mechanistically, SA positive macrophages suppress CCN3, here, CCN3, which has been known as a negative regulator of CD36, thereby enhancing the CD36 expression in hepatocytes. Okay, so with that, I would like to acknowledge my collaborator, Dr. Bin Gao at the National Institutes of Health. So we recently published a paper in Journal of Clinical Investigation, and Dr. Bin Gao and I shared the corresponding authorship, and a graduate student in our lab, Yeonsu Kim and Yukun Kwan, shared the first authorship. Thank you very much for your attention.